Welcome to this explainer. Today, we're jumping straight into the absolute marvel that is biotechnology. If you've ever wondered how scientists literally manipulate the building blocks of life to create life-saving medicines or engineer drought-resistant crops, you are in the perfect place. We're going to break down this complex chapter by treating it like a guided tour of a microscopic factory assembly line. We're talking the tools, the machinery, and the actual step-by-step processes that make biological manufacturing a reality.
To get us started, let's quickly lay down our academic baseline. The European Federation of Biotechnology officially defines this field as the integration of natural science in organisms, cells, parts thereof, and molecular analogs for products and services. I know that's a mouthful, but in simpler terms, it just means we are taking the raw natural genius of the biological world and combining it with human engineering to create some seriously useful stuff.
Here is our factory floor plan for today. 1. Principles of biotechnology. 2. The genetic engineering toolkit. 3. Vectors and competent hosts. 4. The recombinant DNA process. And 5. Scaling up production. Let's get into it.
Section 1. Principles of biotechnology. Think of this entire field as resting on two massive pillars. On one side, we have genetic engineering. This is basically our design phase, where we're going in and literally altering the chemistry of genetic material, like DNA and RNA, so we can introduce it into a host and change its physical traits. But look, a genius design is absolutely nothing without a good manufacturing process, right? That is where the second pillar, bioprocess engineering, comes in. This is all about maintaining a strictly sterile factory floor so we can mass-produce our engineered microbes without any random contamination getting in the way.
Moving right along to Section 2, the genetic engineering toolkit.
Before we introduce our first big tool, I have a quick question for you. Do you know what a palindrome is? In language, it's a word that reads the exact same forward and backward, like malleolum. Well, DNA actually has palindromes too. These are spots where the sequence of base pairs reads identically on both strands if you're reading them in the same direction. Keep that in your back pocket because it's absolutely crucial for how our next tool works.
Okay, meet our first piece of equipment, the restriction enzyme. You can literally think of these as molecular scissors. These enzymes scan the DNA, and when they hit their specific palindromic sequence, they lock on and cut both strands. But what's really clever is they usually cut just a little bit off-center. This leaves these overhanging single-stranded stretches that we call sticky ends. And that stickiness is a total game changer. It acts just like molecular Velcro, making it super easy for another enzyme, DNA ligase, to paste a completely new piece of DNA right into that slot. So, we've chopped up our DNA, right? But how do we actually find the specific piece we need? Well, we use a neat trick called agarose gel electrophoresis. Because DNA fragments are naturally negatively charged, we just load them into a gel matrix and turn on an electric field. The fragments get pulled toward the positive side. The gel acts exactly like a microscopic sieve, meaning the smaller pieces squeeze through much faster and travel further than the big clunky ones. It's a brilliantly simple way to perfectly sort and isolate the gene we want. Which brings us to Section 3, Vectors and Competent Hosts.
Now we need a way to actually deliver our engineered DNA into the factory, the host cell. For that, we use vectors, which are usually plasmids or bacteriophages acting as microscopic delivery trucks. But a vector needs 3 specific features to do its job. First, an origin of replication, ensuring that whatever alien DNA we attach to it will actually multiply inside the host. Second, selectable markers, like antibiotic resistance, which flag down the tiny handful of cells that successfully took up our DNA. And third, cloning sites. Ideally, just one clean spot where our molecular scissors can cut, letting us slip our DNA in without completely destroying the vector itself.
But here's the catch. Getting that delivery truck inside the host is notoriously difficult. DNA is hydrophilic. It loves water, so it strongly resists passing through the fat-based membrane of a cell. We actually have to force the bacteria to take it up by making them competent. Sometimes we do this by soaking them in calcium and shocking them with heat. If we're dealing with animal cells, we might use tiny needles for microinjection. And for plant cells, we literally fire the DNA into them using high-velocity gold particles in a ballistic device literally called a gene gun. Seriously, how cool is that?
All right, Section 4, the recombinant DNA process.
It is time to officially fire up the assembly line. This manufacturing process follows 4 very precise, non-negotiable steps. Step 1, isolating the pure DNA. Step 2, fragmentation,
chopping it down to size with our restriction enzymes. Step 3, amplification, which is making millions of copies of our specific gene. And step 4, insertion, getting that recombinant DNA safely into our host organism to finally kick off production.
Let's zoom in on step 1. DNA is locked deep inside the cell, swimming around with RNA, proteins, and lipids. We have to essentially dissolve the cell open with enzymes and then aggressively clean away all that extra junk. The magic happens when we add chilled ethanol to this purified mix. The pure DNA actually precipitates out of the liquid, clumping together into what looks like fine, suspended white threads.
And this is where it gets incredibly tactile. You can actually physically extract that purified genetic code, a step known as spooling. You can literally just reach into the vial with a glass rod and twirl up those threads of DNA. It is a wildly tangible moment, pulling the very code of life right out of a biological soup.
For step 3, amplification, we rely on a total game changer called PCR, or polymerase chain reaction. This allows us to synthesize massive amounts of our gene in a test tube. It's basically a rapid-fire temperature cycle. First, denaturation, heating it up to separate the DNA strands. Next, annealing, cooling it down to attach little chemical primers. And finally, extension, where an enzyme builds the new strands. Repeat that cycle enough times, and you can amplify one tiny microscopic segment of DNA 1 billion times over in practically no time at all.
And that brings us to our final part, Section 5, Scaling Up Production. Look, you can't supply the world's medicinal needs using tiny lab flasks. We have to go massive. Enter the bioreactors. These are huge, high-tech vats that can handle anywhere from 100 to 1,000 liters of culture. Think of a bioreactor as the ultimate factory floor. It doesn't just hold the cells, it meticulously controls everything. The temperature, pH, oxygen, nutrients, providing the absolute perfect environment for our engineered microbes to turn out huge quantities of our product.
But hold on. Just because the bioreactor is done churning doesn't mean we're finished. Before any of this hits a pharmacy shelf, the raw product has to go through downstream processing. This is a rigorous series of separation and purification steps. It's absolutely vital because it ensures that our final biotechnological product, whether that's an antibiotic, a vaccine, or an enzyme, is completely pure, perfectly formulated, properly tested, and 100% safe for the market.
So let's zoom out for a second. We've cracked the tools, the molecular scissors, the vectors, the massive bioreactors. We have literally mastered the microscopic machinery of life to the point where we can program biology to manufacture life-saving solutions. Knowing all this, what unimaginable biological solutions will humanity engineer next? It is an incredibly exciting frontier. Thank you so much for joining me on this explainer, and keep questioning the science shaping our world.